This is a production of Cornell University.
Okay, yeah, so I'm here today to talk about some recent work I've been working on in epistasis and allopolyploids. Okay, so in your typical hybrid production, at some point you have some alleles. Some alleles that are floating around in an open pollinated population, and then at some point, either intentionally or unintentionally, they get separated from one another, and you end up with different heterotic groups. And then when those heterotic groups are brought back together, you end up with a hybrid, and you've established heterozygosity at essentially all loci in the genome, right? Okay, so now you've got these allelic interactions, which we think are what is important, is part of what is important for hybrid vigor in these progeny. And so, excuse me. And so I like to think of allopolyploids as hybrids. And the reason why is because you start off with a common ancestor that has some set of alleles, okay? And then through gene mutation and evolution, this thing speciates into two species, A and B, and then that are still more or less related to each other, but hybridization between them becomes relatively rare. But when it does happen, you bring both of these genomes together into the same organism, and you've established essentially heterozygosity at all of the loci. Okay, so based on this, maybe we have some homeoallelic interactions. Okay, and these may be what's important for why wheat is so wild, why a lot of our crop species, including wheat, are so widely adapted. Okay, so brief reminder of epistasis. Epistasis is the interaction between two loci. And these loci can have, I'm showing two loci B and C here. I'm showing for the inbred case. Because a lot of allopolyploids are instantly sexually isolated, they usually cannot continue to outcross after that hybridization event, so they have to self in order to continue on. But what's cool about that is they end up fixing all of these allelic interactions, right? So you maintain allelic diversity across your subgenomes while preserving your genotype by selfing. Okay, and so you can have two loci, B and C. Perhaps these are homeoallelic, right? So one is on the A genome, one on the B genome. And there's various models we could have for how these genes might have an interaction. So A will be the effect on the phenotype, okay? And you have the classic multiplicative interaction, which you see often. You may also have like a duplicate factor interaction, which is analogous to two genes that have essentially the same function. And so if you have a functional copy of one or the other, you get the effect, and otherwise you don't. And similarly, we have complementarity, which is like two genes in a pathway. You need two functional alleles in order to get the result. But you'll notice that in inbreds, and this isn't true for hybrids, or I'm sorry, heterozygous populations, you'll notice that the complementary is essentially just the mathematical inverse of the duplicate factor. Okay, so I'm not going to talk about the complementary anymore, but I will revisit the duplicate factor. And so I would like to propose another way to look at this epistatic problem that is related to subfunctionalization. Okay, so the idea is there is some haplotype A in the ancestor, okay, and then through time and mutation, you get a change in this allele A, or you can allow these alleles to diverge in their function. They don't have to, but you can allow them to. Okay, and then when you bring them back into the same organism, like in an allopolyploid, you essentially get the sum of their effects times some divergence from their initial value. Okay, so the idea here is you'll notice that when D equals 1, they have diverged such that they have completely new function. And so they no longer perform the same function within the organism. And when D equals 0.5 and these are equal, they essentially have not diverged in their function. Okay, and everything in between and past, right? So, my subfunctionalization epistatic model actually covers subfunctionalization, which is when D is in between 0.5 and 1. But it allows for a completely additive model. as well as for something that's like overdominance, when D is greater than 1, there's something about having the sum, having two things is greater than the sum of its parts, which you could come up with all kinds of fun biological reasons for. And then, of course, when D equals 0, this ends up being the inverse of the multiplicative, which is kind of complicated, and I'm not going to go into it. OK, so from an ordinary least squares perspective, you can think of this problem as you have an additive effect for the B allele, an additive effect for the C allele, and then an interaction. Okay? And what's cool is in inbreds, if they're truly inbred, you can code all of the markers as 0 or 1. Okay? So you either have the B allele or you don't have the B allele. And then the interaction is just the product of those. And so here I've got so this I'm showing like marker scores. But then you've also got, this is just like a design matrix, right? And then this would be the additive effect of B, C, and then the interaction effect as beta. And what's nice about this is because you only have zeros and ones, this nasty matrix algebra looking thing here really simplifies into a much more simple equation that is just a function of allele frequencies, which is super neat. And is a nice property of having zeros and ones. And so once we can get the expected value of y's here, we can actually figure out what the answer to this, what the expected answer to these betas are. And what that's going to do is it's going to allow me to figure out what the interaction is, or what the relationship is between the estimated additive effects and their interaction effect. OK, so there's some math that goes in there. I'm not going to show all of that. But too far. So you'll see that here for the multiplicative epistatic model, essentially the additive effects will end up being 0, and the interaction is non-zero. In the duplicate factor, we have the additive effects being positive or negatively related to the interaction effect. And similarly, in my subfunctionalization, or what I'm calling subfunctionalization, when D is less than 1, there will be a negative relationship between the additive effects and the interaction. And when D is greater than 1, there will be a positive relationship between the additive effects and the interaction. OK. Unfortunately, if you want to test all pairs of epistatic loci, you run into I'm going to put this thing down because I'm using it too much. You run into a multiple testing problem. Here, even with a modest amount of markers, like 6,000, you have about 17 million tests. Okay? And one of the things I can do is we can use some properties of linear mixed models and epistatic genetic covariances, where so this looks disgusting, but the most important thing is you have some genetic effect, okay? And then you can have an epistatic effect that is simply the Hadamard product of two separate genetic covariance matrices, okay? And with the 0-1 coding, this all works out very beautifully, and it drops out such that this Hadamard product is exactly equal to the covariance that you would calculate based on all of these pairwise interactions. Okay, so then you can estimate a genetic effect for their interaction, and you can estimate a genetic effect for the additive portion. You can also transform these back into marker effects, but that's really ugly because you have to make a matrix of this size. So I haven't actually done that, but I'm utilizing this property to keep from having to deal with 17 million. Okay, and so then I'm just going to look at what the relative relationship between the additive effects is and the interaction. Okay, so this is the classic way. You think of a phenotype as some function of the genetics, and what I'm going to do is I want to find groups of markers that make sense to test for epistatic interactions, okay? But I want a relatively small number of those. That way I don't have 17 million of them, right? So what I do is I might pick, say, 2 homologous chromosome arms, right? Chromosome 1A, the short arm of chromosome 1A and the short arm of chromosome 1B, right? And I can pluck those out. And so I can model the rest of the genomic effect, account for it, while I look at the additive effect of each chromosome arm, as well as their interaction. So, and then I use a series of nested likelihood ratio tests to test whether or not these additive effects are greater than, or have a non-zero variance, and then to test whether or not the interaction has a non-zero variance. And once I establish that both of those things are true, okay, then I can look at the relationship between their additive effects and their interaction. Okay, and so I did actually test this on real data. I have also simulated it, but I think the real data example is much more interesting. And so relatively larger population, 1,200 lines, 7,500 observations. I also have about 6,000 markers that have been pretty heavily filtered for missingness and minor allele frequency. I impute this, and then I set the heterozygous calls to the major allele, because I need everything to be zeros and ones, right? So I can't have a heterozygous call. And so essentially these small 2% of heterozygous calls that I get, I just set to the major allele. And then importantly, because of this epistatic model I was showing you, effectively I've made the assumption that having that being functional allows you to increase the value, say yield, okay? So then I have to phase the markers by their effect. And so I just run a GWAS, and then I just multiply, and then I just have all of the positive effect alleles be a 1 and the negative effect alleles be a 0. Okay. And so, yeah, here's the result from that. So here I have, out of 42 homologous chromosome arm pairs, I test all 42 of them, and I find 8 of them that have significant additive effects as well as significant interactions. And you'll notice I find so here I've colored these. The red indicates that there was a negative relationship between the additive effect and the interaction. The blue indicates a positive relationship between the additive effect and the interaction. Okay, so the blue corresponds like D greater than 1, or like that overdominance. And the red corresponds to more of a subfunctionalization. And then these black ones are a couple that I found that have significant interaction effects, but don't necessarily have a significant correlation to the additive effects. So I don't know what's going on with those ones. But you'll notice that I have tested these at an alpha of p equal, or alpha of 0.05. Remember, I did not correct for multiple. This has not been corrected for multiple testing. And when I do, I basically lose these things, which isn't super surprising, but kind of frustrating for me. These multi-kernel models get kind of difficult to fit, because I've got this whole, I've got this big genome, right, and it's having an effect on yield. And then I'm taking one tiny little piece out of it, and I'm setting it aside, and I'm asking, what is the variance that you explain? And then what is the variance that your interactions explain? And I think that I'm just getting so small that it's hard to find really significant differences. But I did do this for actually all 861 chromosome pairs, regardless of whether or not they are homologous or not. And I do find signal all over the genome. Particularly within the B genome, there's a lot of signal. There's also a lot of signal between the A and the B, but not necessarily on what we believe are homologous regions. So yeah, and this is at a Bonferroni of 10 to the minus 5 roughly. So and the bulk of them tend to show something that looks like this subfunctionality, which I really I guess I haven't really had enough chance to digest these results from why I would expect this. But it is interesting to see. Okay, so in conclusion, epistatic interactions do appear important, but I'm finding them mostly within and across the A and B genomes and not necessarily homologous regions. I do keep one of those homologous interactions, but one out of, I don't know, 20 or whatever it was is arguably maybe not the most significant thing in the world. And it may be because the signal is being drowned by having multiple interactions. Because there's such large pieces of chromatin I'm testing. So one way I'm thinking about circumventing this moving forward is, now that we actually have a reference genome, I plan on scanning the genome for triplicate genes. So basically you find one gene in the A genome, one in the B, one in the D, and then you take the marker that's nearest those, and then basically I would just use the multiplicative products of those markers. And so then I would be drastically reducing the number of interactions I'm testing, and therefore I may pick up a lot more signal. And with that, I would just like to thank my committee, the Yannick Sorrells group, and the small grains field crew, which I could not have all of this data without. Okay, thank you. Yeah, but you're still making, so you're still actually making, see, this is the problem. If you've already done the test, right? You already asked whether or not their additive is significant, right? So then you have to take that into account. Right, so you have to have a correction for the fact that you said, okay, now that I'm saying that their additive is significant, then, but yeah, I mean, you could do that. You could just condition on it and not worry about it. I mean, right. 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 Yeah. And right. Right. Did. Okay. Yeah. Yeah. Okay. Here.
It looks like you have more interactions between non-homeologists, chromosomes, I mean, is that?
There's also a lot more of them, but yes.
Is that representative of, like, I guess the first question, is it more proportionally, or is it just like they're both about the same? And then the second one is that representative of, like, pre-allopolyploidy chromosome duplications? And lots of things have really ancient chromosome duplications. Is that about symphony between 1B and 2B? Or
It could be. I mean, I could speculate a lot on this too, especially since there's also been translocations that have happened since, right? And so I have not been specifically careful about saying, okay, this region is exactly syntenic to that region on these 3 genomes. I basically made the assumption that there's more or less gene synteny from chromosome to chromosome, which is arguably a pretty rough assumption to make. I just wanted to do this genome scan to see if I could even see it, right? Yeah. But yeah, it's possible that you could have ancient duplications doing this. I would expect them to have diverged to the point of being either completely additive or fixed by that point, though. In fact, I'm concerned that part of the reason I don't see this is actually because wheat is too old of a hybrid, even though it's not that old. So it was the last hybridization event happened like 10,000 years ago. So, Christine.
Somewhat relatedly, I was wondering when you go to the triplicate gene type of question, do you have enough data or information available to look at functional copies, or whether there's been some pseudo-genes? Like, is there a way to test only if you have 3 genes and they're all functional?
Right.
But if you're actually coding a functional protein.
So I think if you wanted to ask that question, the cool way to do it would be to do like exome capture. And then you could grab all of those sequences. The problem is that my markers aren't in genes necessarily, right? But wheat has like really large regions of chromatin or LD blocks. So I'm hoping that I get that. A better response to your question would be, yes, you could find all of those, but then how do you decide whether or not they're different? Because they're certainly not identical by descent, right? And they might be identical by state in their amino acid sequence, but without sequencing like the transcriptome, you wouldn't even have a wonderful idea of how close they were. Anyway.
All right, let's thank Nick.
Okay, thanks.
This has been a production of Cornell University, on the web at cornell.edu.